Or
Yes.
they will wait, then
Yeah, I mean, there's one person, but I think
okay.
she just
Should
wanted
we
to.
wait?
I think we can get started.
Yeah? Okay. So hi everyone, my name is Vaso Makeraki. Chris, I'm associate professor here in the Department of Genetic Medicine. Welcome. I know a few of you. I will meet the rest of you. So today is the lecture today is an introduction to stem cells and human-induced pluripotent stem cells, their applications, and the emphasis would be like brain and neurodegenerative diseases. Okay, so I would like to give first an introduction about stem cells, what they are. Okay, it's like a small brief background. So, what are stem cells and how we develop human embryonic stem cells? So we start with the sperm, the oocyte, the sperm, the fertilization of this, and the generation of the fertilized egg, which is called the zygote. So then after multiple divisions, we will come here and we will have this formation which is called blastocyst. The main cell of interest are these cells, which is about 200 cells, and they call the inner mass cells. So we can isolate these inner mass cells, and we can place them in a dish and give all the trophic factors they need, and they can grow and they can multiply. And this is how the first isolation of human embryonic stem cells was done. I have here few images. They are neurons and they are building cardiomyocytes because this is what we can do with stem cells. So, the stem cells they have two characteristics, the embryonic stem cells, okay? So the first one, they can proliferate, they can self-renew, okay, indefinitely, actually. And the second main characteristic is that they have the potential to become every single cell type of the human body. So they can give rise to mesoderm, ectoderm, and neuroderm. And that's why we call them that they are pluripotent, because they have the multiple potential to become every single cell type. The first person that isolated the human embryonic stem cells was Thomson as you can see here, 1998, in the University of Wisconsin, and that was a huge discovery. However, we were facing after the significant discovery and their extended application. Still, there were some disadvantages of using these cells, which are major thing. They have significant ethical issues. What does mean significant ethical issues? If we go back, it's all has to do with this process. So the ethical part is because these blastocysts, these states where we can isolate the cells from, they are becoming from in vitro fertilization clinics. So in vitro fertilization clinics, you isolate eggs from the woman. So in vitro, you do the fertilization with the sperm, and you generate the fertilized egg or the zygote, that then you have the blastocyst. So actually, you can produce multiple blastocysts in vitro fertilization clinics. Some of them, they will transplant it back to the woman, okay? But the rest of them, so they can, instead of being destroyed, they can give, of course, with all the normal and legal procedure, consent forms and everything, they will be given to science for research. So there is the thought of the argument that the human being, the human life, starts in this stage, in the zygote. So when you take the inner cell mass from this blastocyst, it means you destroy the blastocyst, so you destroy the human living. So that was the main ethical issue that came up after the use of human embryonic stem cells. And of course, the second one, disadvantage or the limitation, is you cannot generate patient-derived stem cells. That if you have patients and you know it, you can isolate these stem cells. That's why some years later, actually in 2006, Senior Yamanaka, a Japanese researcher, came up with a cutting-edge and amazing technology, which is called reprogramming. What he did, and I'm going to say this in 2 sentences, but you can imagine how much work needed to develop this methodology. Actually, I'm going to send you this paper, which is a landmark paper for the discovery and generation of iPSCs, induced pluripotent stem cells. So what he did actually, as you can see here, so he isolated fibroblasts. He took some mouse fibroblasts, clean fibroblasts, and he found out that you can use only 4 genes. You can overexpress only 4 genes, and you can transform this mouse skin fibroblast back to the mouse embryonic stem cells. So as I said, it looks simple. I said this in like 4 factors done, but this, he tried many different factors, about 24, and different combinations of factors to be able to come up with these ones, okay? He did this in 2006 and with a mouse, and in 2007, 1 year later, he managed to reprogram human fibroblasts and generate what we now call iPSCs, which is called induced pluripotent stem cells, because we induce them, okay? So, and for this big discovery, he got the Nobel Prize in 2012, which is again an amazing accomplishment, only 6 years, after this reprogramming methodology, and it opened a huge window in the biology today and what we can do with these cells. So now we can have induced pluripotent stem cells in the lab that they mimic the human embryonic, but without using the human embryo. And this is how they look like. So this is a colony. It's not a cell. This is a colony of human-induced pluripotent stem cells. These are the teeniest, 10 micrometers, which are small cells, but they are extremely powerful cells. So when we were first isolating this, and we were making these induced pluripotent stem cells, so we were growing, we were using a substrate. This, you can see how different they are from this, and how it's a fibroblast. These are mouse fibroblasts, okay? So the idea was that these ones, these iPSCs, they needed all the trophic factors that they can get from the mouse fibroblast in order to be able to stain undifferentiated, okay? So first we were growing these iPSCs in the fibroblast. After years, we finally we will be able to grow these iPS without the use of this fibroblast, but only with specific substrate, with specific media, without any other factors. But it took years to develop all of these methodologies. So this is schematic representation of how you can generate here these iPSCs in the retrievers in your lab, and these are actually images of this process. So you can start specifically. This one is something that we have done in my lab many years ago when we were using human fibroblasts. So you could buy human fibroblasts even from stem cell banks or stem cell company, or sorry, not stem cell company, cell banks or cell companies. So we could buy human fibroblasts from Alzheimer's disease patients that they had a specific mutation, which is called A246. I will tell you later about this. It's a presenilin mutation that causes familial Alzheimer's disease. And then you can use this reprogramming method using episomal plasmids. Episomal plasmids, so you will be producing these exogenous genes in the cells. And after keep pulsating these cells, finally, after many days, almost a month, you can have nice, beautiful colors. So you can see, mostly I added this figure so that you will be able to see how you change what you see in the lab and how the morphology changes, what are the stages, okay? So you go from human fibroblasts to human-induced pluripotent stem cells, okay? However, some years later, after using fibroblasts, they found different methodologies because you can imagine that more and more people, they start working with stem cells, which means that all the protocols have been developing stage by stage. So now we are able to use a little bit of blood of a patient. It can be, it doesn't matter the age, it can be 10-year-old or it can be 80-year-old, okay? And we can take like 8 ml of blood and we can isolate the peripheral blood monocytes and we can use the same methodology and have from blood differentiated cells of an 80-year-old person his embryonic stem cells when he was an embryo in our days. And since, as we said, these are the potential, they are pluripotent, they can give rise to neurons, they can give rise to muscles, organ, plant cells, bone cells. It depends on what we want to study. So different labs, different stem cell labs, depending on the disease that they study, they have a different cell focus that they can differentiate. This process called also differentiation from an iPS cells to a specific differentiated cell. Okay. It doesn't mean that people they don't use fibroblasts, skin fibroblasts now. They do, but the peripheral blood is much easier because with the skin fibroblasts it's more painful to have a biopsy. You need a topical anesthetic. Instead, you can have 8 ml of blood and you can do the iPSS. And this is something that we are doing in the lab in a daily basis, actually.
Are iPSC generation not affected by the accumulation of mutations over years? So if like these cells in this patient come from an octogenarian, the Yamanaka factors are not impacted by any of those?
No, you actually eliminated the genetic factors. And if they have a mutation, not because they are 80-year-old. So if they have a mutation like when they call their Alzheimer's disease, for example, familiar, of course it stays in that you will see after the reprogram because it's in the gene. But this accumulation of mutations, we don't see in the iPSS. You can see accumulation of extra mutation during when you passage the cells for many passages. And at some point you can have like more than 100 passages. You can have different mutations, different order translocations, but that's why it's not good for your experiment to start working with this iPSS and you try to have low passage iPSS to start your experiment. So what are the advantages of this? So one of the main advantages is first we can have iPSS that they are patient specific because as I said we can take blood from different patients and we can make their own iPSS. So we can model the human disease in vitro and theoretically now for some diseases they are doing, you can make the once the cell type that is missing or for example if there is dopaminergic neurons, if someone has Parkinson's disease and there is a certain type specific loss like dopaminergic neurons so that they are lost during defective during Parkinson's disease, theoretically you can have the blood of this patient, you can generate his iPS cells, differentiate to dopaminergic neurons, and then go back to the patient and transplant. Theoretically, it will happen in a few years. In some diseases, for some diseases, especially in the retina, they have done already. They are doing this now. So talking about first, like precision medicine. So this is the main hypothesis about, not the main hypothesis, the main idea of precision medicine. So when we talk about precision medicine, what it is. So let's say you have a population, a heterogeneous population, and for most neurological diseases, we have, of course, a heterogeneous population, which means a population with different backgrounds. So the standard method is to give the same treatment to all of them. The different backgrounds are shown here with the different colors. Standard approach is to give the same treatment to all the patients, but this is only partially effective, as you can see here, okay? So when we talk about precision medicine, the ideal thing is to be able to define the subpopulations of people that can react to a specific treatment. So you will be able to give the specific treatment to each of these subgroup of patients. And then, of course, the treatment would be very effective. So while we are talking about, I said that the focus will be neurodegenerative diseases, brain and neurodegenerative diseases. I want to give you this as an example of this precision medicine. Okay, so precision medicine and Alzheimer's disease. So just a brief background about Alzheimer's disease. Okay, I believe everyone knows, but just a reminder. So this is a slide that Dr. Tom Price, one of the biggest guys actually in the field of Alzheimer's, he was also here in neuropathology in Johns Hopkins. So he put up this slide where you can see all the pathology that you can find in the brain in the patient with Alzheimer's disease. So you can see this big formation here, which is amyloid formation, which is A-beta. I guess you have heard about A-beta protein that causes the amyloid aggregates, and you have the plaques, one of the hormones of Alzheimer's disease pathology. So you have these big aggregates of this amyloid, and you have synaptic disconnection and the neurodegeneration. You have microglia creation that after you have the inflammation there, they can be activated. And you also have this other cell pathology, which is called tangles, here inside the neurons. And these are forms of, these are aggregates of a protein which is called phospho-tau. It's a protein that, tau is a protein that in the cytoskeletal protein. When it gets phosphorylated in Alzheimer's disease, it gives these aggregates, which is called the neurofibrillary tangles, that they can cause the death of the neurons. So this is the all the pathology that we can see in the brain, but we don't know the sequence. We don't know what comes first, what comes second, and how each of these pathology influence each other, okay? So until now, over the last decades, this is the main hypothesis how Alzheimer's disease was caused. So you have this A-beta oligomers, which is called A-beta 142 that I showed you, that they can form the blast. So this, the misprocessing of A-beta and the generation of these 142 oligomers can induce the formation of the tangles that can cause neuronal death. Then the whole neuronal sequence, they will collapse, and then you have the symptoms of Alzheimer's disease. So this was called amyloid hypothesis. Most of the research was driven all the decades with this hypothesis. So based on this hypothesis, we assume that Alzheimer's disease is a senile disease entity based on this. also, and that's why we don't have treatments until now, and also because until now we were using animal models, okay? Most other Drosophila or mouse models or yeast models. So someone was saying, an expert in Alzheimer's disease, they were saying we managed to treat mice, all the mice with Alzheimer's disease, but we don't have any treatment for humans because we were using drugs to see, or they were going to clinical trials because they had very good results in mice, but then they were tested in humans, we didn't have any data. So now we believe that maybe it's a time to use some human models. Okay? And not only this, maybe we need to have a shift in the paradigm that Alzheimer's disease is not a single disease entity. So because now we know that maybe there are upstream processes that even though still tau and beta plaques are still the main pathological feature in Alzheimer's disease, but now we have a shift in the paradigm because we know that there are many other upstream, other processes, other conditions like genetics or trauma that they can influence this whole path, or other processes like inflammation, oxidative stress, that they can have an effect in the process, in the whole process of this whole, in the whole process of the disease, starting from the oligomers until the symptoms of the disease. So how we, so that's why you can see all these multiple errors instead of one in each stage, meaning that there are many different pathways that they are involved, and these pathways are different in each person. So how we intervene in each of these stages depends on what pathway the patient is following. Again, that's why we have this in drug failure. Because maybe you have heard about the last year, there are a couple of, there's a big news about a couple of drugs that they are in Alzheimer's disease, that there is not a cure, but what they manage is to just slow the disease. Even though they can eliminate some of the plaques of the brain, still there is no cure about the disease. So they just slow the whole process. And since 2003, only 98, we have 98 drug failure at the clinical trials, what I told you before. And this is because of the patient heterogeneity. So when the disease develops in each patient, the disease progression is different in each individual. The symptoms, the severity of the symptoms are different, and the response to different treatments are different, is different in each patient. So the bottom line is, and that's why we're talking about precision medicine in Alzheimer's disease as well, is because it's not Alzheimer's disease is not a single cell entity, a single disease entity. It means that different people, they have different backgrounds so that they can influence the disease progression. So what we need to do is to define the biological subtypes of Alzheimer's disease. So in our lab, our precision medicine approach for this disease is to make iPS cells from Alzheimer's disease patients. So the lab is, my lab is part of the Precision Medicine Center of Excellence in Alzheimer's disease here at Johns Hopkins. You may know that there are multiple precision medicine centers for different diseases. Okay, one of them, a new one, is in Alzheimer's disease because we have this idea about the heterogeneity of the disease. So what we are doing, so patients that they come to the Alzheimer's disease research center here at Hopkins, and they are enrolled, they give consent forms, and we can take a little bit of their blood. Of course, all the diagnostic data are in the Athg system. We don't have access, but we collaborate with the doctors and physicians that they have access to them. We only need some ml of the blood. We make in the lab their own IV cells, and then we differentiate different cell types. Alzheimer's disease, of course, is a brain disease, so we need brain cell types that we need to differentiate. And depending on the question that we need to answer, we differentiate to cortical neurons, serotonergic, astrocytes, organoids, different types of this. And the idea is to use this not only to study disease pathology in our Athg, but also to use them as platforms for drug screening, so that we will be able to see which of the pathways that I was telling you about are involved in each individual, and how we can develop a specific treatment for each of the individuals that we have his own stem cells in the lab. So this is the schematic. What is we have what we are doing. So recruit healthy and patients, make their own personalized iPS cells, generate the iPS cells, and then after you generate the iPS cells, this culture that I showed you, then you have all these iPS cells that have to pass a quality control, which means they have to be, you have to make sure that the cells that you generate, they resemble the human embryonic stem cells, which means the karyotype, they have a good karyotype, they have you need different real-time PCR, flow cytometry, different methodologies to see that they express all the pluripotency markers, and also that they have, we call it spontaneous differentiation, which means that they have the main characteristic, which is the potential to differentiate in all three lineages, mesoderm, ectoderm, and endoderm. And this is the ultimate test that these cells has to pass and make them become and make them valid as stem cells, so that they can differentiate to ectoderm, ectoderm, mesoderm, and endoderm. So in the lab, we have done more than 100 iPS lines from different Alzheimer's disease patients and healthy individuals, Alzheimer's disease patients of different age, different sex, and with a different genotype, ApoE genotype, I will explain to you. And we have done all this full characterization. And again, then, since we have this stem cell pack in the lab, we want to start answering some questions. So, going back to this scheme, we need to differentiate, for example, first to neurons, and then to other different cell types. So when we say differentiate into different cell types, it means that we are using the knowledge that we have so far from the biology, developmental biology, about what genes, proteins overexpressed or are on or off during the brain development. So we are trying to mimic the brain development in the disease. That's what human embryonic stem cells are doing, and so we try to add different factors, and here is the neural tube formation in the embryo, where we know that the neural tube during the brain development, it starts with the development of the notochord in during the day 8 of the day 20 of embryogenesis, and it has this characteristic. This is a schematic of the neural tube. It has this characteristic formation when you do a section of the neural tube, and we call this characteristic formation neural rosette, because this is like a beautiful rosette. So the neural tube is the one that will give the brain and the spinal cord. Okay, so that's what we are trying to mimic in the disk when we start differentiating iPS cells to neurons, and this is iPS cells differentiating cells to neural tubes. You can see how beautiful they resemble the neural tube formation, and this is the beginning of protocols and other people that they are working with neurological diseases what they are trying to do when they start differentiating. So this is the schematic of with very few information there, because we are the different factors of how you start from IPA cells, and then you go IPA cells all the way to neuronal differentiation. And this is the actual images. So starting IPA cells, then you have these formations, we call it embryoid bodies, where you start with IPA cells, and then there is a methodology which is called force aggregation. So these cells, they start from these embryoid bodies that they mimic the blastocyst that I showed you, this formation. Okay. And then you take these embryoid bodies, and of course they are floating, and then we add different factors that we push the cells towards the neuroectodermal lineage. That's what we're doing. If other people, they want to work with muscles, they push the cells, they add different transcription factors in the media, and they push towards the different lineage. Okay, so we want the neuroectoderm. So then we attach these little balls here. They attach to a substrate, and they form these beautiful neural rosettes. That these neural rosettes are the neuroepithelial cells. Neuroepithelial cells, or neural progenitor cells, if you see, are the cells that they immature cells, okay? That they can give rise to both neurons and glia, okay? Depending what you are going to add and what factors they get and what inputs they get, they can develop to neurons in the brain and the glia astrocytes in the brain, okay? So we can isolate these neuroepithelial cells. Here is a beautiful neural rosettes. And actually we can manually, under the microscope, isolate these neuroepithelial cells, and we can grow them in Petri discs here. And we can do stainings. I guess everyone knows what are immunostainings? Yes? Yes, perfect. Okay. And we can use different markers for neuroepithelial cells. This is a validation that we have we have good differentiation efficiency and pure neuroepithelial cultures. And then these neuroepithelial cells, we push them, we add different factors so that they will be able to become neurons. And we can see here, they can form beautiful neuronal secrets in our blade. And then we can do different characterization for these neuronal cultures, where we can stain with the markers for synaptophysin, for markers for glutamatergic neurons. All these different colors, different stainings are for different markers and different types of neurons. And of course, you want to see if these neurons that you get are functional. So the main functional study is the electrophysiology, where you can see that we have live, active cells that they can talk to each other, and they are beautiful. And most importantly, when you have these nice neurons, we want to see if they have the pathology. How do we see the pathology? These are 2D neuronal cultures, which means that they are attached. And what we have seen with ELISA that you can measure the secreted proteins A beta, the amyloid proteins in the supernatant. It's amyloid beta 40 and amyloid beta 42. And the ratio of these two proteins is a characteristic hallmark feature for Alzheimer's disease patient. Okay, so when we want to see that, say that how we can measure, or if a person has an Alzheimer's disease, one of the measurements is this ratio here. So we do this for this neuronal culture, take the supernatant and do this. Again, these are 2D cultures, okay? So these are pure neurons, 2D cultures. But if you want to study the disease in the best possible way, you need to have a way to mimic the human brain where the disease develops. Okay, and what is the best way? The best way is to make the organoids. The organoids here are 3D cultures. How can you study why you need these 3D cultures? When we say, does everyone heard about organoids, 3D cultures? Perfect. So you want to study, you want to see the pathology, the real pathology. You don't just want to see proteins in the supernatant of cells. You want to see the aggregation, which means the plaques. You want to see the tangles that I told you. You want to see the neuroinflammation. That's how, if you have a model that mostly resembles the human brain, maybe you can have the better answers for the questions that you have, and you can resemble the brain and the pathology in the best possible way. Before going to the organized Alzheimer's disease, I would like first to give you a very brief background about the organoid. Okay, so and why the organoids are important and how everything started with the organoids that we have the science of organoids. I said that we don't have, not only for Alzheimer's disease, but for other diseases as well, treatments yet for these diseases, because the models that we are using are not human, and they cannot resemble brain development. Even though we have very good information so many years, and we have taken advantage and have done all this amazing science with the mice and the mice brain development, still there are major, of course, differences, as you can see, not only for the timing of the brain development between the mice and the human, as you can see here, but the structure, the cell types, everything. So one of the main also problems is of studying the brain development is that the human brain develops in the uterus, so we cannot go there, we cannot intervene, we cannot see how the brain develops, and these are the most critical steps. We don't know, for example, which one is the first neuron that fired, how the first neural circuit developed. That's why we are hoping that with the human organoids, brain organoids, we will be able to study all these questions at the beginning of the brain development. And also another important thing is that the human neurological diseases, either they are neurodevelopmental or neurodegenerative diseases, they are very complex. They have a complex genetic. Most of them, they are not only caused by one mutation or from a small group of genes. Most of them, they are, the cause of these diseases are because of different variants that they spread all over big areas of the human genome. So we want to model the human genome, not the mouse genome. And that's why also it's necessary to start working with organoids.
This is really cool. My question is, with Alzheimer's, like I know that there's of course like a strong genetic component, but a part of it is environmental. So when you're taking like fibroblasts from patients and turning them to iPSCs, do those cells still have the same environmental exposures that the patient, like what I'm wondering is how do they develop these plaques if there's still like an environmental component that
then you have to introduce other plaques, or you have to introduce the environmental components afterwards. You want to have the important thing is to have the genome of the patient, okay? And you have their own, and one of the main questions that we always get, the people that we are talking, they are studying neurodegenerative diseases, as you mentioned, with iPSCs, because iPSCs is the first stages of the embryonic development. Okay, they resemble the fetus embryonic, fetus neural cells. Neurodegenerative diseases, it's after 80 years. 80 years we see the symptoms, actually it starts many years before. How can you study neurodegenerative diseases with iPSCs? So the idea is that you resemble part of the pathology, not everything. You cannot have everything going on in the patient, an 80-year-old patient, in your neurons. Having, be able to have plaques, I will show you later, be able to have plaques or tangles characteristics of egg pathology in your list, it's a big thing, because then you can start testing drugs, how you can eliminate. For example, this is an example case, this pathology. So as I said, you cannot totally mimic the whole pathology of one person has, but at least you can mimic some of the pathology. Like the environmental factors, of course, that the person was exposed, you cannot, but you can add different, test different environmental factors that a person could be exposed exogenously and see what is the effect on the cells that we have. Does it make sense? To some extent, I know, it's to some extent.
I was going to also ask, like, depending on, let's just say, is there any work that's been done where, like, you take this wild type iPSCs, not from a patient with Alzheimer's, but you modify that ApoE4 locus, and then you expose it to stress and see if those develop
all the time.
as well?
Yes, you can have, you can introduce mutations. You can change, you can make isogenic lines, and you don't necessarily need, but you can have whatever IPS line, and then start introducing a mutation. But it's not going to be from a specific patient that you have the clinical data of this patient. You know what, if he has this genotype, the other genotype, what are the variants, and study this person, and study the brain of the patient. Definitely, there are models like this. You introduce a mutation that's familiar, for example, Alzheimer's disease, it can be familiar and sporadic. Familiar Alzheimer's disease is because of different mutations, okay? So yes, you can take again as a line, introduce a mutation. But then you study the effect of the mutation, nothing more than this, okay? So, going back to the organoids, that's what we hope from the organoids, and that's what we've seen from the organoids, what they can teach us. It's how the human,
I was curious from like the opposite perspective. Has anyone ever grown like healthy organoids and then induced tau tangles or amyloid plaques and then seen some of the impact?
Oh yes, yeah. This is easy. Not easy. Nothing is easy. Of what I'm saying, okay? Nothing is easy. Nothing is fast. Okay. It needs training. It needs time. It needs effort. And these experiments will take some time to develop. It's not like a cancer cell where they grow in one day. If you want to have an organoid, it depends again which time point you want. It's 1 month. I will show you. 1 month, 2 months, 6 months, and it can go on. So having this, it's again a similar question. Having this, yes, you can introduce some A-beta peptides and they can form. But again, you cannot have a clinical data. It depends again on the question. Okay, you can have it targets or aggregates, and you can test a drug how you can eliminate the plaques, but you cannot have an organoid from a patient, and so that you have the clinical data from the patient and study other pathways that can be involved in your organ. Okay. Again, depends on the patient. So you can use organoids for human treatment, so the disease pathology, genetics, and as platforms for drug screening. So, brief background about how, from the 2D cultures, we ended up to the 3D. Okay, so this is another landmark paper. It's from a Japanese guy, Sasai, okay, who actually, what was the question and what he saw? How we can make these brain organoids? It's very easy to say now, but it's very difficult to be done in the humanities. So what he saw, and this is an optic cup, this is an eye, it's not, this will give an eye, it's not a brain, okay? So what he did is actually that the cells, the human embryonic stem cells, he did this with human embryonic stem cells back and then. You can see a beautiful formation of an optic cup. They can self-organize. So if you took some human embryonic stem cells, put them in a specific medium, and they show that they can self-organize and they can give the optic cup. The most amazing thing was that not only they can form an optic cup, they can self-organize and they can self-pattern it, but they can have the cells that they are in the eye and in a specific structure, actually the structure that you can see in the eye, in the human eye. So you can have our outer neural layer, inner neural layer, ganglion cell layer. Okay, and you can see this beautiful structure in a section of this optic cup, which means that these cells, they have an implicit information and that can be, to some extent, they can self-organize. Okay. So the question was, can this also be done with the brain? Can also the cells self-organize and have a cortex and become cortex and have a nice structure of the cortex? And actually, some years later, see these Lagman papers, he showed that you can have beautiful cortex. Again, took this embryonic stem cells, did this quick aggregation that I showed you, and make embryonic bodies like this, and he showed that you can have a beautiful cortex, and if you cut, if you section this, and you can stain them with specific markers for brain cells, not only you can see the brain cells, but also you can see a beautiful structure like during the human brain development. And it's funny, not funny actually, but the thing is that in these papers, you will not see the word organoids. Okay, so this was many years ago, they were talking about structure and how it can be organized. Then later, a few years later, the first paper which mentioned organoids was Lancaster and from Europe. So as soon as he mentioned organoids, the word organoids, that became a thing. But this guy was the first one that managed to make these 3D cultures, and these are the first ones who had from 2D to go to 3D. Okay?
Were these cortical layers induced by transcription factors that were in the
Yes,
Okay.
yeah. Well, so there are many, since then, okay, there are many different milestones during the formation of organoids. I'm going just to say a couple of them, so that because you will hear the term organoid, you will hear the term spheroid or assembloid. Okay, so there are different. So we have the guide. We have guided and unguided formation of brain organoids. When we talk about unguided differentiation, it means that the self-organized, you just leave them there, you don't add anything, and then what you have a cerebral organoid that it can have different regions, cells from different regions of the brain and structure from the different regions, even an optic cup. So the cells are organized. Remember, these are from all human embryonic stem cells or human-induced pluripotent stem cells, and then you have different areas. When you talk about guided, it means that in the cell culture system, you add different pattern signals, okay, different trophic factors, transcription factors, so that you can guide the cells to become different regions, specific regions of the brain. So then, like, you can have a midbrain organoid, hypothalamic organoid, thalamic, depending, again mimicking the nature. That's the most amazing thing with IPSS, that you can add different factors and you can overexpress or downregulate genes, and depending on these pathways, you can have specific brain regions. Yes?
Is there any difference between like embryonic stem cells and iPSCs in their ability to self-organize?
No. That's the beauty of the iPSS. So it's so all these experiments are with iPSS. The first one with optical was with human embryonic, if I remember correctly, but these ones are all iPSS. Now, most of the people, almost all the stem cell scientists, they work with iPSCs because of all the issues that I was telling you, and because now you can have patient-specific study the disease. Okay, so, and if I go back here, what I wanted just to mention is that you have the assembloids. So you can go here and you can put in the same list cerebellar or hippocampal organoids with the thalamic organoid, hypothalamic, you can, or a midbrain organoid, you can put all together and they start attached to each other and they start connecting and they can start extending neurons between each other. And now people, they can add different labs, they are starting to add even the eyes, the opticals. So you can start an optical, you can go with, try to bring together and with an thalamic organ, for example, okay? So how long can you, what's going on with this in vitro culture? First of all, do they have all these brain cell types? And how long can you give them? Can you grow them in culture? So, and how do they mimic also the brain development? So they mimic to some extent, of course, the brain development, as I showed you, the cell types. And you can grow these organoids. This is from a lab, it's called, it's Arlota, the PI's name is Arlota, but recently in a conference he mentioned that she can grow the organoids, his organoids in culture for 7 years. So they go from postdoctoral fellow, one the postdoctoral fellow leaves, the next one gets the organoids, so go from postdoctoral fellow to postdoctoral fellow, okay? And during 9 months, they can see that it's a little bit necrotic inside, but other than this, it's about 5 millimeters, okay, the biggest that they have seen in size, but it can grow lively for 7 years. Now, I don't know for how long they will keep in culture. Yeah, this is how they look like, okay, in culture. So it's pretty amazing because it looks like a small balls, but when you start sectioning, you can see all these beautiful cultures. Okay, this is from a different lab, and then you can see how homogeneous they are. And this means that you can have hundreds or thousands of these, depending how much money the lab said, and so you can have many, okay, because it's also very expensive type of research. And this is what we are doing in the lab. So actually, again, an abstract of the protocol that we are doing. We start again with human iPSCs, make these beautiful embryonic bodies, but instead of attaching to have the neural rosette that I showed you, now we are growing these embryonic bodies in these flasks. They are spinning. Maybe you can see the magnet. They are in magnetic field, so it goes like this. And you can grow them there for months. And so that's why they call 3D cultures, okay? And you can see with time that you have this beautiful formation, which are the neuroepithelia formation, which means they form neural tube. And you have this, it depends how much, how long you can keep this, they start forming. Here is how they look like in the incubator. These are the flasks. And why the people have found out that it's better to use the spinning flask, and it's always, first of all, they don't attach. It keeps them for attaching. You can grow up to 100 in this flask, but also by doing this, with this rotation, you have a better oxygen diffusion and nutrient diffusion that goes inside this ball. And you come almost every day and you see that they keep growing. Okay, and here is how they look. And here is a graphic. Some more images, how they look like, and this how they form this beautiful neural process. You see here, you see here. I don't know, is it clear to you? How? For me, it is because I see this all the time. But I hope you can see, beautiful neural mosaics. With this spatial microscope. And I just want to give you an idea. With this information and here of a neural mosaic. So, this is how they look outside. What you can do with them. So actually you can treat them like a human brain almost. This is how they look like under the stereoscope. And then what I mean by you can treat them like human brain, like if you are in a human pathology lab and they take autopsy brains, they cut them, section, and they do this famous Athg staining, and where you can see the nucleus and the cytoplasm. So you can do the same with organoids. You can use a microdome and you can have thin slices, frozen slices of like 10, minimum 10 micrometers thick, and you can do any type of staining that you want, depending on what you want to do. So you can do Athg staining, you can do immunostaining, fluorescent staining with different markers. As you can see here, this is the neural rosette, this is neuroepithelial and neural rosette formation, and stain with different markers of different cell types. MAP2 is a marker that people that are working with neurons is most common marker for labeled neurons. Okay, W of course is the nuclear. So here is less 3D image how if you take with the microscope a very fast 3D image that it shows the neural rosette, how they form and how you can say. So the structure, it's all about the structure. It's not only the cells that they have, but also how they structure inside. So here is, this is an illustration of how the human, from a real human embryo, human neural development, during the human neural development, the cortical development looks like, where you can see that the neuroepithelial cells and the neurons, they can form beautiful structures, and they can have the subventricular zone, interventricular zone, outer ventricular zone. And what we wanted to see is in our organoids, can you see same or similar structure to the cells in the organoid, organized in specific layers, or they just all over the place. And we did staining with different markers for neuroepithelial cells, the most famous are SOX2 and PAX6 here, and we can see that yes, they can form these beautiful structures, the beautiful layers, where we can see the neuroepithelial cells in and the neurons and then the neuroepithelial cells, different layers. They can also form what we call these structures that all the people that are working with the brain development, they say that they mimic the ventricular or subventricular zone here. These are PAX6 neuroepithelial cells. And we can see if we stain with other markers, these are for outer glial cells that we see in the brain development, that we have this typical, these neuroepithelial cells, they have this typical formation. They have apical processes, they don't have basal, which is very typical for human, during the human brain development. And if we leave the organoids to, this was in a 30-day organoid, if we leave the organoid to mature more, this is a human brain, okay, where you can see different layers of neurons, and we see two sections in two map organoid for different types of neurons. We have the early-born neurons, the late-born neurons, and we have different markers to stain these different types of neurons. For example, CTI2 is for early-born neurons, BRM2 is for late-born neurons. Again, we can see that they form beautiful layers. And we can be doing staining with to see if they form neural sequence with different markers. For example, GLUT1, which is a glutamine transporter 1 marker. It's a marker with on the synaptic vesicles. We want to see how mature these organoids they are, and or synaptophysin. And we show that they are mature and they form synapses. Again, this is after 2 months organoid. And also, if we leave the organoids for more than 2 months, we not only see brain cells, but we or neuroepithelial cells, but we also see astrocytes staining with GFP and oligodendrocytes that takes even longer to make, staining with myelin-based protein here. Okay. And if you go and do single cell analysis, you see all these different types of cells, depending again in which stage you will get the organoid. This is from molecular uterine stem cells. So you can see glutamatergic neurons, which is most of them glutamatergic, which are the first neurons, type of neurons that you get during the development. You still have progenitor cells that then they will become astrocytes or other types of neurons, glial cells. Okay, so actually what I showed you is like in this organoid you have structure, they resemble very close to some extent the molecular and cellular biology of the human brain. Okay, so how can you use? This is only 2 of many examples of how organoids have been used. Just to give you a very brief idea how what are their applications and how they can be used to find treatments. Okay, so you may remember the Zika virus outbreak in Brazil some years ago. So they used the organoids to find the drug. Okay, so what they did is they took the Zika virus, they isolated the Zika virus, and they used human brain organoids, as you can see here, and they infected the human brain organoids. And if you remember, the babies, the mothers, if they were infected with Zika virus, the babies, they were born with neurodevelopmental disorders of the brain. I think it was microencephaly. And why it was this? So smaller brain, because when they did the experiment with the brain organoid, they saw that the Zika virus, it was infecting the neuroepithelial cells, okay? The cells that they will give neurons, they will become neurons or astrocytes. So they were infecting this type of population, which means they were dying, which means that the cortical plate, as you can see here, diffuse cortical plate and dying neural processes, so that the brain was smaller, okay? And they used this method, and they treated the organoids to see with different types of drugs, this one, and for protection and for treatment, and they saw that they can restore the human brain. They can prevent this death of the neural progenitor cells. This is one paradigm. Of course, you can find all the papers about this. And another cool thing that we can do with organoids and cancer, okay? So you can have a brain organoids and you can introduce glioblastoma cells. Glioblastoma cells, as you can see here, glioblastoma cells, they are fluorescent. They are labeled with a GFP protein, and they are fluorescent, so they can form glioblastoma tumors in and out of the surface of the brain organoid. So the cool thing is that glioblastoma cells are very difficult to treat, of course, and there are no drugs for this, but if you have patient-specific cells, iPS cells, you can make the organoids and you can test different drugs specific for each person that they can kill the glioblastoma cells, but they can leave the brain organoid intact. So we talk, it's a paradigm of precision medicine here. Okay. So, going back to Alzheimer's disease, after this small parenthesis, again, why we see, besides the fact, why we want to test these organoids, we want to study Alzheimer's disease with the organoids and not only 2D cultures. Because I think you are convinced by now that they resemble the human brain, they model the human organogenesis, they mimic the cell environment. But for Alzheimer's disease, it's important to have organoids because that's the only way to have A beta aggregates plaques. Okay? Why? Because first of all, you can grow the organoids for 6 months, 1 year, so you can grow for a long time. So you cannot do this with the 2D cultures, only neurons. They will die after 2 months. Okay? But with organoids, you can have it, so you give them the time to be able to develop this pathophysiology of Alzheimer's disease. So first of all, you can have them for a long period of times, so they can age. And second, you allow the beta aggregates to form and to remain stable because you have a small ball of cells. The cells secrete this A beta that they form the aggregates and they stay inside. If you have 2D neurons, if the cells, you cannot see these aggregates because every day or every 2 days you just remove the supernatant and you just add new egg. You don't have the time to form this. So this gives us the opportunity, the organoids, to study to model better the disease pathology. And of course, I will mention later, you can have other cell types that they play major role in Alzheimer's disease. So here is a brain organoid from a familiar Alzheimer's disease patient. It has a presenilin-1 mutation that it can cause the A beta aggregates. And we have it's a section. This is a higher magnification of, not from this, from another organoid, but a higher magnification. We have stained 48, so 48 is a very popular antibody that in the field of Alzheimer's disease, where they use to detect A beta plaques in human autopsy brains. So we are using the same markers, of course, in the human organoid. So where you can detect other soluble or A beta aggregates. Okay, so we see that this pathology takes more than 100 days to develop. Okay, it's not that you will see them after 50 days of organoids or 1 month. So you need to wait a lot of time to develop this pathology. So we have A beta aggregates. I don't like to say plaques because they're not plaques, but they are A beta aggregates. Plaques are. Okay, and this is a 3D imaging of this, the same staining of an A beta aggregate, how it looks like in our brain organoids. And not only this, you remember I told you that it's not only A beta plaques, there are also aggregates from phospho-tau, when this tau protein that helps the microtubules to be stabilized gets phosphorylated in Alzheimer's disease, then it forms this phospho-tau aggregates, which is called TAPIs, and we use a different antibody, TFH1, to see these tangle formations. So we have the organoids with the tangle, we have the organoids with pathology. So here I want to go back, not go back, but I mentioned that we can give you a paradigm how you can use these organoids as drug screening platforms and what you can do with them. I mentioned that with the pathology you can test different drugs. For example, you can test these drugs that they have been developed now to eliminate the plaques or the target with the plaques. So you can test this in the organoids. But you can do also other things. So here is the paradigm, an example, where we have generated in the lab cell type specific organoid, a hindbrain organoid, or a brain region, I would say, specific organoid. So, and why we were doing this? Because we know that patients in Alzheimer's disease, okay, some of the patients in Alzheimer's disease, not all of them, they can develop what we call neuropsychiatric symptoms. These symptoms, or NPS, that's how we call it. This is agitation, depression, psychosis, okay? Some of them you also see in schizophrenia, some of them, okay? Let's focus on AD. So what we believe is that these symptoms is because there is a problem, there is a dysfunction in the serotonergic system, in this area, this system where the serotonin is the serotonin released from specific type of neurons that's called serotonergic. There is a dysfunction in the or misregulation in the release or in the uptake. Neurons release serotonin and they can be uptaken from the neurons next to it. So until now, there are some treatments. As you can see, it's called SSRIs. These are FDA approved drugs, very common drugs for this type of symptoms. And these are the ones that they have been giving to patients with these symptoms. But what they see is that they don't work for all the patients, okay? And they work with a different, there's a moderation, they're different in the effect of the drug in different patients, okay? So there is a patient heterogeneity and clinical variability in the timing, the type of the symptoms, the neuropsychiatric symptoms, and also the effect of the drug in these different patients. So it works in some of them, but it doesn't work to others. Okay. So the idea is why it works in some, what is how the serotonergic system work in some patients, and how they react to this drug, and in some patients, how in other patients. So a precision medicine approach, based on what we're doing, would be to take blood from healthy individuals, take blood from Alzheimer's disease patients with these symptoms, and take blood from Alzheimer's disease patients without the symptoms, and make the IPSS. And that we have been doing from in the lab. We have been taking blood from all over the United States. They can sip it, and we can make the IPSS. And then, again, we have done all the quality control, okay, and we have generated the brain region-specific organoids, which are the high-brain organoids that they will be rich for serotonergic neurons, because that's what we want to test, okay? Since these are the ones that are misregulated during these symptoms. We have used different protocols to generate these organoids. We call it high-brain organoids. This is already published, okay? We have done stainings with specific markers. 5-HT is a specific marker for serotonergic system, and we can see a subset of cells mimicking what, because not all the neurons, of course, in the brain are serotonergic, only about 30% are serotonergic. So we can see a subset of cells having serotonergic neurons. We have done, as it was expected, we have done real-time PCR just for the characterization to see how the efficiency of the differentiation, so using different markers for different neuronal markers, but also different markers for serotonergic neurons that they express only these neurons. This is part of the characterization. So then, after we have done, and we have proved that we have done the system, and have these neurons are dysfunctional, which means, do they secrete serotonin? That's the most important thing. Because if they don't secrete serotonin, then we cannot use them as drug screening platforms. And yes, we have seen that they can secrete serotonin in the superdata. We use ELISA methodology to measure this. And as you can see here, and then, since we saw that they secrete serotonin, we have functional serotonergic neurons, we want to test the pharmaceutical efficacy of different drugs. In this specific case, a serotonin, an SSRI drug which is called escitalopram. The way that this drug works is actually it blocks the serotonin receptor, so it doesn't allow serotonin to be reuptaken from the cell, so you have increase of serotonin in this extracellular environment. Okay? So actually, we measure the serotonin secreted in the cells after we add the drug. We have done different tests regarding, first of all, the toxicity of the drug to find the right dose of the drug and the different time points, and we saw what is the very nice thing to see is that we can use these organoids to see the effect of the drug, because we have seen that we have used different SSL concentrations. So we have seen that the response to the drug is drug dose dependent. As you can see, they respond with different after different concentrations, and this is after 1 hour treatment with the drug. Okay, and the very nice thing to see is what we expected, and what we see in the patients is what we see in the organoids, that there is a variability. Some, for example, two of them, the healthy one healthy, here two, and this one, they don't respond to the drug, but there is a remarkable 2 to 3-fold increase in the extracellular serotonin. Does it make sense? Okay. So this is a sense of variability that we get, the variability that we see in the human patients. You can say that we have a sense of variability, but of course, we need to do many more IPS lines from many more IPS patients so that we'll be able to say that we have this group of patients respond differently to the drug. Because remember, that was the original idea of the precision medicine, to be able to define biological subtypes that will respond to a different drug, to a specific drug, and give the specific drug to this specific biological subgroup. And this is now what we are doing. We are doing proteomic analysis where we have made all these organoids with and without treatment, triplicates, do proteomics, transcriptomics, and see all the effects and analyze now the data. So, what time is it? How much time do I still have? Because
12 minutes.
12 minutes. Okay, okay. Because I can go for hours. Okay, so
I wanted to ask a question about the last slide. Is there any clinical study where they've given like patients with Alzheimer's Lexapro and seen their response? Escitalopram?
Oh, escitalopram, yes.
Mm-hmm.
There is a clinical. See the latest last month's Nature Medicine paper.
Oh, okay.
Yes.
So
It's actually from Johns Hopkins.
Oh,
It's a
okay.
clinical trial.
And from a standpoint of drug development, does it seem like wise to have like you have your cohort of patients and maybe using the same method identify responders and non-responders?
Yes, that's the idea. So that's the idea of why to do the organoids or IPS from patients. Because, for example, this one is from patients. These patients have been in a clinical study with this drug. Okay, so do we see the same results with IPSS, the same effect in the organoids with the clinical response that the patients have, and connect the data that we have from the organoids with the clinical data, with the clinical outcome? Because then we can say, instead of testing mice, test drugs in the human organoids if there is good correspondence. Okay, and this would be faster, and this would be definitely more relevant because it's human cells. So the idea is this, that's the real beauty. The clinical data, patients that have been taking a drug, organoids from the same patients that we give the drug and compare the clinical data. And then we can just have organoids from patients and test different drugs. Yes?
So one day, do you foresee that it will be possible to sort of extract the features about patients or which groups they're in from the organoids to the point where you would be able to predict, someone's group or subtype without having to grow an organoid from them? Does that make sense? Like eventually down the line.
Without doing organoid, how?
I'm saying, say you grow these organoids and eventually you find that
You grow the organoids.
What?
You said you grow them.
Yes, so
yes.
you grow organoids and you find several subtypes of Alzheimer's disease. Say you find 10 subtypes of Alzheimer's disease.
Yes.
And you know that
subgroups, yes.
patients in these subgroups need these drugs, these drugs,
Yeah,
these
yeah.
drugs. Eventually, do you think you could find, genetic or clinical features that would allow you to say you get a new patient you've never seen before?
Oh, this is like
You could-
AI that
Well, I'm
they
saying
can
you get a
train
new
models.
patient. Like, do you think you will always have to grow organoids to treat the patients, or could you eventually learn enough to where using other information you've correlated with prior organoids, you could say, we don't need to grow an organoid for you because we know
Yes,
other
yes,
information you're
yes.
in
So it's
subgroup,
like
whatever.
using all the data from the hundreds or thousands of organoids and train AI models to see if this, from the data that you get from the organoids, and say, oh, this subtype has this characteristic. If you go to the person, I don't know how you can do it without doing. You need to do some studies in the new patient that will come. How can you train to?
Yeah, but the things you would do in the new patient don't necessarily need to be the organoid, right? They can
Yes.
be the genetic testing.
Yeah, oh, yes, some, yes. For some tests, yes, definitely. So some use some of the data from the organoids, yes. So that's the other idea, that all these you can grow hundreds, thousands, millions maybe, okay. And you can train them, you can have models that you can train and see this characteristic means that for some, no. So,
Sorry, one more question. Do the organoids mimic what pharmacogenetics predicts in terms of like serotonin response to these SSRIs?
yes, to some extent. To some extent. We need to do many, many more studies. Okay? But to some extent, yes, they can. You mean the clinical data that we see? Oh, no.
Just from like maybe non-impactful like SNPs, but changes to like the receptors themselves, whether that's trafficking of the receptor
Yes.
or
So it's mostly the pathways.
Yeah.
Yes. That's another beauty of the organoids. Okay. So we call them mini brains, but they are not brains. Okay. You will see mini brains. We are always hesitate to go outside and say mini brains because people that they are not in this field in the biology, there's always this: are they going to develop consciousness or not? We just need to explain that no. Also, the important thing to remember is that it cannot become a brain because after a while it gets necrotic in the center. So we say that we can grow for 7 years, but in the center, even after a few months, you can see a big hole because they don't have vascularization, there is no blood, there is no oxygen. So you have a ball of cells, they're outside, they get the oxygen and the nutrients, but the inside it doesn't. Okay, so it cannot, never say never, but now in the field how things are, they cannot become a brain. Okay, but the whole different labs, biotechnology, biomedical engineering, we are working on how you can incorporate blood vessels. Okay, so there are different ways. You can make an endothelial organoid, which is only about endothelial cells that they form the blood vessels, and then you can take this endothelial organoid and you can fuse it. Actually, we had submitted a paper right now. Take the brain organoid, take an endothelial organoid, you fuse it, and then you have one organoid with endothelial vessels, blood vessels inside. Of course, no blood, but what you can do with this, you can study the interactions of endothelial cells with neural cells, okay? And you can have like blood-brain barrier models and all the stuff. And also, I said you can have many different setups, many different neurons, many different, you have astrocytes, oligodendrocytes, but you don't have another setup which are the microglia. Okay, you don't have the immune system. And in Alzheimer's disease, the immune system, it's very important because not only in Alzheimer's, but also in many other diseases, you need to have neuroinflammation because the there are different interactions of the microglia, the immune system of the brain, with the different cell types. For example, what I want to say. So this is just a. They just say. Okay, so this is a laser point. This is a culture of microglia, immune cells. So they just have the laser point there, and you can see that they are crawling because there is some damage there. They are crawling. The microglia is immune system. They are crawling around this, and they are moving there. Okay, so they become activated after this damage. So the microglia is the immune cells of the brain. So you cannot, when you develop the organoids, the way that I told you, the organoids are neural cells, it's neuroectoderm, it's ectoderm. The microglia, they develop in the yolk sac, in the human embryo, sorry, they develop in the yolk sac as macrophages, then they migrate to the neural tube during the brain development, and then the blood-brain barrier forms a barrier, and they close in the neural, in the brain, and then there they mature to the mature microglia. So they start in the yolk sac, which means they are different lineages. Brain, neural cells are ectoderm, these are mesoderm, okay? So that's why when you make the organoids and you push them towards ectodermal lineage, we cannot have, the microglia, they are not developing this, okay? Only one start, they say, oh yeah, we saw this. Okay, so what do? You make microglia in parallel with your organoids, okay? Oh, just before saying this, what is the function? They are good and bad function, okay, depending on the states and depending on the conditions. So what they are doing is actually they are pruning the neurons, they are pruning the synapses so you don't have over the generation of many synapses. So they are pruning, they are forming the neural circuits. Okay, they in such a way they also are involved in synaptic plasticity, but they also, since they are the immune system, they can have in Alzheimer's disease specifically, they have two different roles. They have the protective one, where when you see blood, you can see also microglia crawling, and they can go to phagocytose the blood and clear the blood, but also they can be activated from an external damage or from an external, or from these conditions, and they can be activated, and they can cause inflammation from the secretion of cytokines, okay? So we want this here. We don't want this because neuroinflammation means also neurodegeneration. Okay, they kill it. Okay. So what we are doing in the lab, us and others, is like we develop again microglia side by side with the organoids, either to study that by themselves, okay, or to incorporate. So again mimicking the nature, again mimicking what's going on in the human embryo. So what we are doing is now we want to develop the yolk sac in the petri dish so that they will secrete the microglia precursors, the immature microglia. Again, starting from my PSS, now attach yolk sac formation and these cystic formations in the petri dish, they can secrete millions of microglia precursors in the supernatant. Okay? And this is how they look around when they are, these are the precursors, immature microglia that they are floating. They have this around safe, but then when you attach them, and you can start adding the different factors, they can mature to microglia, that you can also do co-cultures, 2D co-cultures with neurons. Here you may be able to see the neurons. All these are fibers that you can see are the actions and the drugs from neurons, and you can see the microglia on top of them. Okay? So when you do this, again, they have to pass a quality control to make sure that they are nice microglia, they express all the factors, so we do the quality control with flow cytometry, where we can see that they express 90 to 99% these factors, CD45, CD11, that they are microglia factors. I'm going very fast because of time. You do the stainings, again, with specific markers. All these are 2D. And then you want to do, okay, we have microglia, are they functional, or are they just sitting there doing nothing? So two main functions of microglia is the phagocytosis. When we see they have an external intruder, they crawling, they phagocytose, and again, inflammation, so they secrete cytokines. And these are important for the Alzheimer's disease starting with phagocytosis. But in order to be able to see that they have this functionality, what you can do is you can buy microbeads, latex microbeads, that they have a fluorescent dye, that fluorescence only when the microglia will go at phagocytosing bead, and the bead will be in the lysosomal compartment in an acidic environment, so then you have a green fluorescent. So this is a glyphosate. So the green means that they have been phagocytosed, these beads, from the microglia. So they are functional, they found the beads, they phagocytose, and they have the fluorescent. And also when you do an activation with LPS, liposaccharide, which is a very common, it's, you know what LPS is? It's in the bacteria, gram-negative bacteria that you usually use to cause neuroinflammation, cytokine secretion, so you can see that they can, you can see that this is before, this is after, before and after, that they can secrete different types of cytokines, okay? All this in a cell culture media. And here in the co-culture, what you see in a slow motion, it moves slow. It's amazing because if you see them under the microscope, you think that everything is, they don't do anything, okay? But if you do the live imaging, these are cells, microglia, from a cell line that is fluorescently labeled. So you can generate red microglia. So if you do the live imaging, you can see them crawling in neurons, and they can change their morphology, how they move, okay? And of course, going back to the organoids, in order to make an even better model, you want to incorporate the microglia and have an immune system in your brain that you want to study. So what we do is we have the organoids, and we take this microglia, we do co-culture, meaning we throw some microglia in the co-culture, and we can see even after a few days, the microglia, they go towards the organoids. At the beginning, they are in the surface, and then they start being inside. Again, this microglia from a cell line, from a red fluorescent cell line, so when we did an imaging, you can see how they look. Some of them in the surface at the beginning, then they go inside. And when we do sectioning of these organoids and stainings with markers specifically for the microglia, you can see all these nice dots, okay? Again, that's an example of how we can try to make better models. Better models means better disease pathology, resembling better answers for our questions, okay? So just to finish, just to summarize, this is what I showed you at the beginning, what we are doing in the lab, the precision medicine approach that we have for Alzheimer's disease. It's pretty much the same for many other diseases. Okay, and you can use these 3D cultures or 3D cultures. You can do genome editing. You already mentioned genome editing. We are doing transcriptomics, mass spectrometry imaging. You can use them in many different ways, apply different methodologies. And another type of research we are doing is called extracellular biopsy. This is for another another lecture. Extracellular vesicles that they secrete. These are vesicles secreted from each cell type of the human body. It's a very cutting edge field in the biology now. We knew them for years, especially for cancer, but only the last years there is a lot of focus because at the beginning they thought that these vesicles, teeny tiny vesicles, 50 to 100 micrometers in diameter, they thought that it was a way how the cell was getting rid of all the garbage, but now we know that there are many different proteins, DNA, RNA, lipids in these vessels, and this is the way one the role is it's in cell-cell communication. So they secrete from one cell, they can be uptaken from another cell. So there is a hypothesis, or we are testing a hypothesis that maybe a disease can be transferred in this way. We have done studies where we have seen that these vesicles secreted from neurons or from Alzheimer's disease organoids, they have A-related proteins inside. Yes, I'm done. Okay, that's it. Okay, thank you so much. I hope you have an idea. I can talk for hours. Actually, I want to emphasize that these organoids, I focus on the brain organoids because what we are doing in the lab, but same type of research and the same concepts are for different type of organoids. You can have heteroids, okay? You can have lung organoids. You can have different types of organoids depending on the lab and what are the diseases that they are working on and the questions. And you can have multiple type of organoids in microchips, where you have a brain organoid, a lung organoid, okay, a limb, and you can try to make a small human person. Okay, so thank you. Sorry if it was too much, but that's it. Okay, if you have any questions, if you want to see organoids,